Target identification in cell cycles is a need of time because it helps us to identify different targets which can be used for drug discovery mechanism and which are used for removal of cancerous cells and then generation of different drugs for different diseases. Now what is the current state of time that there are very few cell targets are available which are based on mechanism based anti-cancer drugs. Now there is a need to identify new drug targets based on this cell cycle methodology which can be used for finding different new targets for drug discovery pipeline. So for that purpose, a common organism which is very relevant to human beings is Drosophila melanogaster, which is used as a organism used for studying different mechanisms related to target identification. If we go into some details of target identifications, we should know that number one, what is a cancer? Basically, cancer cell is a cell basically which has some abnormal activities due to some mutation inside the genes. So if there are two types of these mutations, they can be in single gene or in multiple genes or multigenic type things. So if they are multigenic, then their targets or to work on those multigenic cells, it would be very tough. So for example, if we take example of human beings, at the moment out of 30,000 human genes, we have identified 287 genes which are responsible for production of cancers. On the other hand, if we talk about that Drosophila melanogaster, it has roughly 197 genes. So what happens is that there is a protein P53 or RB basically which should be worked on to identify different targets for this cell cycle based target identification. So if we have this information about these genes, then we can do genetic manipulation to find out different targets for our drug discovery mechanism. So whenever we talk about these identification technologies or techniques based on the cell cycle, there are three different categories. First one is genome based information. Genome based information which takes information about the genetics or genes about different organisms. For example, if we talk about humans, then we have roughly 30 recessive and 100 dominant oncogenes available. Oncogenes means again which have potential to produce or to move towards cancer. And so what happens is that we want to find out human related genes with the help of insertions, deletions, substitution and rearrangements, so that what will happen that based on these different mechanisms or techniques such as insertion, deletion and so on, we want to find out if some gene can be converted into a cancerous genome or cancerous cell. The other thing is about lethality approach. This lethality approach basically it is a synthetic approach and then it works on multigenic cancers. So what is the idea? It detects difference between gene products of normal and cancerous cell. If I take example of Drosophila, then we have roughly 150 mitotic mutants available related to this lethality approach at current times. The final approach of this target identification based on the cell cycle is disease association. Now what happens in disease association? We want to find out connection or association between diseases and genes. So that is basically based on gene expression profile using microarrays, because there is part of combination or association we want to study in detail with the help of microarrays, which is mechanism to study more genes at the same time. Then we want to modulate the targets so that they work in the normal conditions. And finally what happens is we usually block active sites of enzymes by using might be RB proteins. So by blocking these active sites we make sure that there are no cancerous cells produced.